Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the Deep Dive. We're here to plunge into key research, pull out the vital stuff, and help you get really informed fast. Today, we're tackling a truly revolutionary 2008 paper. It didn't just give us a new lab tool, it fundamentally changed how we actually see our genes. It showed DNA activity is way more dynamic, more surprising than we thought, and things like alternative splicing. Turns out that's like a default feature, not some rare exception.
That's a great way to kick it off. Yeah, we're diving into mapping and quantifying mammalian transcriptomes by RNA-Seq by Mortazavi and colleagues, Nature Methods 2008. This is the paper that really introduced RNA-Seq, and honestly, it reshaped the entire game for studying gene expression. Functional genomics hasn't been the same since.
Okay, so before 2008, before this paper, what did gene expression studies even look like? What were people using back then?
Right. Well, the main players were largely microarrays and Sanger sequence ESTs, express sequence tags. Microarrays seemed great, probe tens of thousands of transcripts at once. But they had some pretty serious drawbacks.
Oh, yeah. Like what were the main headaches for researchers then?
Well, first off, microarrays had a pretty narrow dynamic range. That means they weren't great at accurately measuring transcripts that were, really rare, or the ones that were super abundant. It was tough to get good numbers across the board.
So hearing a whisper and a shout with the same mic, tricky.
Exactly. And they also suffered from cross-hybridization. Similar gene versions, isoforms, could stick to the wrong probes, blurring the picture. That's a big problem if you need precision. And maybe the biggest thing, microarrays needed you to already know the sequence. If it was a new gene, a novel splice variant, invisible.
Wow, okay. So if you hadn't already discovered it, you literally couldn't see it. That sounds like a massive blind spot for discovery.
It really, really was.
Yeah.
Then you had the Sanger EST projects. Now, these were unbiased. They could find new things because they just sequenced whatever RNA was there. But, oh man, they were slow and expensive, just prohibitively so for looking at a whole transcriptome, all the RNA in a cell or tissue.
Right. Couldn't really scale it up for comparing lots of samples or anything.
Not practically, no. So there was this huge unmet need. We needed something that could ideally capture everything known, unknown transcripts, quantify them accurately over a massive range, and crucially, reveal all those unannotated splice junctions, the different ways genes can be put together after they're transcribed. That was key.
And that splicing complexity, that post-transcriptional stuff, is super important for development, for disease, right?
Absolutely vital. But the existing tech, it was like only seeing the very tip of the iceberg. We knew there was complexity, but we couldn't map it comprehensively. We needed something unbiased, high-resolution, quantitative. That's the massive gap RNA-Seq filled.
Okay, I see the problem space clearly now. So, knowing all that, how did RNA-Seq actually step in? How did it offer such a different approach?
Well, the core idea behind RNA-Seq is, I think, quite elegant. It's basically a digital measure of genes, completely different from those older analog methods based on how strongly something sticks to a probe. With RNA-Seq, you directly sequence fragments of the RNA after converting them to cDNA. You're literally counting molecules.
Counting, not just measuring signal strength, like going from fuzzy radio to crisp digital audio. The paper's figure 1 kind of lays out the workflow. Can you walk us through that, starting with the biological stuff?
Sure. So you start with your RNA sample. Typically, they use poly-A selected RNA that targets the messenger RNAs mostly, the ones carrying protein codes. And you isolate it from your tissue. They used mouse brain, liver, skeletal muscle. Good complex tissues.
Right. Got the mRNA from the mouse bits. What's next to get it ready for sequencing?
Okay, so that long RNA gets chopped up, fragmented into smaller, easier to handle pieces. Think like shredding documents. Then these RNA fragments get converted into double-stranded cDNA. DNA is just more stable and works better with the sequencing chemistry.
Okay, cDNA bits. How do you prep those for the actual sequencer machine?
Right. Then you ligate or stick specific DNA sequences called adapters onto the ends of these cDNA fragments. These adapters are like handles. They let the fragments attach to the sequencer's flow cell and also provide starting points for the sequencing reaction itself.
And then comes the massive scale part, right? The parallel sequencing. What did that look like?
Exactly. These adapter ligated fragments get loaded onto a next-generation sequencer. Back then, it was the Illumina genome analyzer state-of-the-art. And this thing just churns out reads. Millions of them. Short sequences representing the ends of those fragments. The paper reported like 41 to 52 million mapped reads per sample. Just an incredible amount of data for the time. Reads were short, though, only 25 base pairs.
Wow. Millions of tiny sequence snippets. And the last step is figuring out where they all came from.
Precisely. That's the computational part. You take all those millions of short reads and map them back to the reference genome, find their original location. And this is where the unbiased discovery power really comes in, because you're just sequencing whatever's there in an unbiased way.
Yeah.
You capture everything. Known genes, unknown genes, weird splice forms. You don't need prior info. You just find what's present.
That sounds incredibly powerful, but, it was a brand new technique. How did they actually prove it worked reliably? How did they validate it against the old methods?
Yeah, that was absolutely critical, and they did a thorough job. Figure 2 in the paper really covers this. First, they showed a fantastic linear range of detection. They used known amounts of RNA standards. And RNA-Seq could accurately quantify these across 5 orders of magnitude. That's huge. Low stuff, high stuff, it can measure it all reliably. A massive improvement.
5 orders. That's like measuring ants and elephants on the same scale accurately.
And they introduced a new way to quantify things too, right? Became a standard.
Yeah.
That's right. RPKM, reads per kilobase of transcript per million mapped reads. Bit of a mouthful, but a really important concept they introduced. Basically, RPKM adjusts the raw read, counts for 2 key things. 1, the length of the gene. Longer genes naturally give more reads, right? And 2, the total sequencing depth of that sample, how many million reads you got overall. So by normalizing for both gene length and library size, RPKM lets you compare expression levels fairly across different genes and different experiments.
Right, so it levels the playing field, like saying, okay, this gene got X reads, but let's adjust because it's super long and we sequence the sample really deeply. Makes comparing things make sense.
Exactly, and they backed it up with solid validation. They compared the RNA-Seq RPKM values to quantitative PCR, qPCR, which was and still is a gold standard for measuring gene expression for specific genes. And they found a really strong correlation, like R over 0.92 across a 1,000-fold concentration range. That showed it was accurate.
Okay, strong correlation with the gold standard. And how did it stack up head-to-head against the microarrays they were trying to replace?
That was the kicker. RNA-Seq actually detected 25% more expressed genes than the standard Affymetrix arrays they compared it to. Just straight up found more stuff. That really hammered home its increased sensitivity and discovery power. It wasn't just a little better, it was uncovering a whole chunk of the transcriptome that arrays were missing.
Okay, so it wasn't just counting better, it was finding more. What else did this new digital map reveal about, like, the basic wiring of our genes? Any big surprises in the data?
Oh yeah, this is where it got really interesting biologically. Sure, over 90% of the reads mapped to known exons, which was good. It showed the method worked. But it was the other reads, the remaining data, that was transformative. It pointed to completely new gene models or revisions to existing ones. Things like unknown promoters, new exons, different 3' untranslated regions, the 3 TARs. Yeah. These are critical regulatory regions. So it wasn't just quantifying, it was refining the very map of the genes themselves.
Wow, literally redrawing parts of the gene map based on the sequence data. That's huge.
It is. And they also found evidence for new candidate microRNA precursors. MicroRNAs are these tiny RNAs that regulate other genes. Really important. Finding new ones opens up whole new areas of biology. But maybe one of the coolest things was how it directly detected RNA splicing.
Right. How did they actually see the splicing events? That seems tricky with short reads.
They looked for reads that spanned across 2 different exons reads that physically crossed an exon boundary. They called them splice crossing reads. Microarrays and older methods couldn't really do this directly. They inferred splicing. RNA-Seq provided direct sequence-level evidence of a splice having happened right there.
Okay, so you see a read that starts in exon 1 and finishes in exon 3. Exon 2 got spliced out.
Pretty much, yeah. And seeing this directly across the whole transcriptome was powerful. And what they found was staggering. Something like 1.45 million distinct splice junctions observed. And the big take-home, alternative splicing was everywhere. It wasn't rare. They found about 3,500 genes showing alternative internal splices. This really drove home that point we started with. Yeah. Alternative splicing isn't the exception. It's a fundamental, common way mammalian genes operate. It's the default.
It's clear this wasn't just another technique. It was a paradigm shift. So what was the magic behind it? The innovation, the teamwork, the vision that made RNA-Seq such a game changer?
It really was a combination of things, kind of a perfect storm. First, the how, the technical innovation. This paper presented the first really complete end-to-end RNA-Seq workflow. From RNA prep to mapping and quantification, it was a full package deal that just blew away existing methods in resolution, scope, and accuracy.
So not just one piece, but the whole pipeline, soup to nuts.
Right. And the algorithmic side was huge. They developed custom software, the RNA-Seq mapper. This thing was clever. It didn't just align reads to the genome. It used a seed and extend approach to specifically find those reads crossing exon boundaries. This allowed de novo splice junctionist discovery, finding new splice sites without needing prior annotation with really good accuracy, like down to 2 base pairs. That was a major computational leap. And as we said, introducing RPKM gave everyone a standard way to quantify and compare results.
Sounds computationally intensive for the time. And who are the players involved? It sounds like it needed serious resources.
Absolutely. It was this amazing synergy, a sort of academia and industry triple play. You had Cold Spring Harbor Lab bringing deep biology expertise, especially in splicing. You had the Broad Institute with their massive sequencing power and bioinformatics infrastructure. And you had Illumina, the sequencer company itself, with engineers tweaking the actual machine chemistry for sequencing cDNA effectively.
Wow, that's a powerhouse combo.
Yeah, and that environment meant incredibly fast cycles. Lab protocols tested immediately on sequencers, software results feeding back to lab adjustments, sometimes within days. It just turbocharged the development from a concept to a usable protocol.
That rapid feedback loop sounds crucial. What about the key people? Any individuals who really stand out?
Definitely. The paper acknowledges key contributions. Barbara Mortazavi, the first author and a grad student then, did critical work optimizing the library prep, like fragment sizes, and importantly, developed the RPKM normalization. Real quantitative insight there. Barbara Wold, a bioinformatician, developed that complex splice-aware mapping software in C, tackling big computational hurdles like memory limits back then. And Barbara Wold, the senior investigator, provided that crucial strategic vision. She saw that unbiased sequencing could replace arrays. She took a risk, championing early access to Illumina's prototypes when the reads were still really short, just 25 bases. Not everyone saw the potential then. It was their mix of, benchwork grit, clever coding, and looking ahead that made it happen.
Incredible story. A blend of persistence, innovation, and foresight. So wrapping this part up, why does this paper still resonate so strongly today? What's its lasting impact?
Well, theoretically, it fundamentally changed how we think about gene expression. It established this digital counting principle. It's not fuzzy signals anymore, it's discrete counts. And it made it undeniable that things like pervasive alternative splicing and antisense transcription are just normal features of our genome, not weird edge cases. That shifted basic biology understanding.
And technologically, what did it set in motion?
Technologically, it gave us the first truly scalable, annotation-independent RNA-Seq workflow. Methods like RPKM, splice mapping variations are still used. It basically began the process of making microarrays obsolete for most expression studies, and the applications just exploded from there. High-res maps of transcriptomes in different tissues, finding cancer-driving gene fusions, paving the way for single-cell RNA-Seq, which is huge now. It also laid groundwork for using RNA in the clinic for diagnostics, looking for splice mutations or fusions as disease markers.
So it really just opened the floodgates for a whole new era. Were there limitations they recognized even then?
Oh, for sure. The short reads were a limitation, making it hard to map in repetitive regions. The poly-A selection missed RNA types without poly-A tails. And initially, they lost strand information, couldn't tell which DNA strand the RNA came from. But honestly, the conceptual shift was the main thing. Moving from hybridization to sequencing was the paradigm change. That shift kick-started constant improvement, longer reads, strand-specific methods, single-cell tech, direct RNA sequencing. It chased all of modern genomics.
Wow. So this deep dive really underscores it. Mortazavi et al. Wasn't just about a new technique. It gave us a fundamentally new, much clearer lens to see the incredibly dynamic world inside our cells, moving from fuzzy analog guesswork to precise digital counting.
Exactly. And connecting to the bigger picture, the legacy is just huge. It sparked a genuine revolution in how we approach gene regulation, disease mechanisms. It opened up so many doors, things we take for granted now, like single-cell RNA-Seq becoming a standard lab tool, trace their lineage right back to this 2008 paper in the groundwork it laid.
Which brings us to a final thought for you, our listener. Given how far sequencing tech has rocketed forward just since 2008, think about it. What other hidden layers of biology might still be waiting to be uncovered? with even better resolution, higher throughput. What secrets are still locked away in the complex dynamic code of our own cells? What might we find next?